Okay, good afternoon, everyone. Thank you all for being here. My name is Mike Connolly, and I'm very excited to share some of the results of my project using low coverage whole genome sequencing or genome skimming to delimit species and assess the population genomics of East Pacific Pocillopora corals. Um, so Pocillopora corals are a really important model species. First, because they have a broad geographic distribution from the Western Indian Ocean to the Eastern Tropical Pacific and are hardy, resilient corals that can survive in marginal habitats. Second, because of this resiliency, Pocillopora corals tolerate captivity better than other species and are easy to culture in aquariums. So we can create small replicate fragments to use in complex manipulative experiments to better understand coral cell biology and microbiology. However, a major problem is that Pocillopora coral taxonomy is still unresolved and species identification is notoriously difficult. So early taxonomists described over 50 species of Pocillopora corals based on morphological traits like branch patterns and polyp arrangements. The problem is that Pocillopora corals demonstrate extreme morphological plasticity, and this makes visual species identification extremely challenging, especially in the field where the same species growing across different environments can present multiple morphologies. So that in the late 1970s, many of the original species names were grouped together and synonymized as ecomorphs of only 21 currently accepted species. However, molecular genetic studies in the past 2 decades have uncovered substantial genetic diversity despite high morphological plasticity, indicating that our current taxonomy does not adequately account for the number of genetically and potentially ecologically distinct Pocillopora species. And so this is the problem as we try to understand how differences in these species biology and ecology can affect ecosystem responses to climate change. So the question I've been interested in throughout my postdoc work is how can we better identify ecologically and evolutionarily distinct Pocillopora species? So to accomplish this, I've been working to apply process-based species delimitation approaches that better resolve evolutionarily discrete coral species lineages. And so in doing this, we're following the unified species concept, which defines species conceptually as separately evolving metapopulation lineages. And in this framework, many lines of evidence can be used to support species delimitation, and different criteria for speciation, such as reproductive isolation and reciprocal monophyly, can emerge at different time points during divergence. The good news in the case of Pocillopora corals is that within the last few years, multiple reference genome assemblies have become available, and their quality and completeness continue to improve. And there's a wealth of Pocillopora genomic data that is growing exponentially. And so recent sequencing of ultra-conserved elements, or UCE loci, from corals collected across the Central Pacific and Indian Oceans allowed this process-based species delimitation framework to identify Pocillopora genomic species hypotheses that are now being used to rectify taxonomic problems in other areas of the world. So for this work, why are we focusing on the Eastern Pacific region? It's a key gap in these efforts, and it's geographically isolated from the world's most biodiverse coral ecosystems, and with a different oceanographic regime that limits the diversity of other coral taxa. And as a result of these relatively recent oceanographic conditions, Pocillopora corals have become the dominant reef framework builders throughout this region and are ecologically critical keystone species. So this research that I'm talking about today was done in collaboration with a network of scientists at the Smithsonian Tropical Research Institute in Panama, the National Museum of Natural History, and research universities in Central and South America. So coral tissue and DNA samples were assembled at the museum from sites in the Gulf of Panama, in Gulf of Chiriquí in Panama, Isla del Coco, Golfo Dulce, and Bahia Culebra in Costa Rica, Isla Gorgona in Colombia, the Galapagos Islands in Ecuador, and Clipperton Atoll offshore of Mexico, for a total of 342 samples that were processed for this low coverage whole genome sequencing or genome skimming approach across 3 sequencing lanes this past year. So our laboratory methods included a DNA extraction and library preparation using a New England Biolabs kit, And then we sequence these samples using Illumina short-read sequencing to a target depth of around 10,150 base pair paired-end reads per sample. I processed the reads from sequencing and ran downstream analyses to extract coral mitochondrial genomes and a key diagnostic species barcode, map reads to a Pocillopora reference genome and called genome-wide SNPs for use in population genetics analyses, and then lastly, assemble contigs using spades to extract these UCE loci from multiple sequence alignment and phylogenetic tree construction. And all of the analyses are available on my GitHub at that URL, Michael T. Connolly, ETP, Pocillopora, GSkim, if you're interested in applying these approaches in your systems. So for the mitochondrial genome analysis, we were targeting a highly variable barcoding locus known as the MTRF. This barcode has been developed as a species diagnostic marker in previous Pocillopora taxonomic revisions and provides a starting point for assessing the species diversity we observe in our East Pacific dataset. So we identified this locus in all of the mitochondrial genome assemblies assembled during the analysis, which is over 95% of our samples with greater than 1 million reads, which highlights this efficiency of mitochondrial, of recovering mitochondrial genomes and barcodes using this low coverage sequencing approach. Next, I had to correct for the presence of naturally occurring clones in the dataset for downstream population genetics analyses. So to do this, we identified genome-wide SNPs, or single nucleotide polymorphisms, and clustered samples on the basis of technical replicates to provide a threshold for within individual similarity. And basically, this analysis identified 229 unique genotypes in the data set, with 46 genotypes having naturally occurring clones that we sampled, and 183 singleton genotypes. So we were able to use this clone-corrected SNP data set to perform principal components analysis that revealed 5 genomic species clusters, which in this principal components plot we're calling clusters 1, 2, 3A, 3B, 4, and then several admixed individuals located only at Isla del Coco that appear to be mixed between clusters 1, 4, and 1, 2, and 4. We were then interested in assigning these genomic species clusters to the previous taxonomy using the genomic species hypotheses from the Central and Central Pacific and Indian Oceans. And so we completed a maximum likelihood phylogenetic analysis of UCE loci from the top 5 samples of each of these clusters, and we're able to assign cluster 1 as Pocillopora effusa, cluster 2 as Pocillopora meandrina, and clusters 3A and 3B formed a monophyletic clade within the Pocillopora grandis species complex, and cluster 4 formed a clade, a similarly monophyletic clade within the Pocillopora vericosa species complex. And so taking this a step further, based on analysis and an examination of the type material from historical taxonomic work of Pocillopora in the region, we see that our field specimens of Pocillopora effusa and meandrina closely match the expected morphologies given the type specimen, whereas in clusters 3A and 3B and cluster 4, their morphology more closely matches the nominal species Pocillopora capitata and Pocillopora lacera, which were described from the Eastern Pacific, namely Oaxaca, Mexico, and Islas Perlas in Panama. And we believe that the genetic divergence we see in the genomic data and their close correspondence to the morphology of these type specimens suggests that there could be a case for revising the taxonomy of Eastern Pacific Pocillopora based on these. Lastly, there were some biogeographic patterns that emerged when we examined which species are present at which locations. The continental sites were dominated by our Pocillopora capitata and lacera lineages. So cluster 3b was dominant along the continent, whereas there was a lineage of Pocillopora capitata 3a that occurred at our offshore sites of Isla del Coco, and the Galapagos, and Clipperton. When we look at the variation between these offshore and continental Pocillopora capitata lineages, we observe very high morphological plasticity within and between the lineages, and a clear signature of genetic differentiation between these populations. When we look at the loci that are the most significant drivers of variation along the principal component, we find some really interesting results as well. The top loci are heat shock protein 14A, matrolin 2, and cell cycle division control protein homolog. And it's possible that these loci are responsible for the differences in the environments that these lineages are inhabiting. So it's just another example of how powerful this data is for analyzing the genomic underpinnings of coral ecology within this region. So for the next steps, we're working to detect more adaptive loci that are under selection between these different Pocillopora species lineages, as well as exploring the patterns of hybridization and intervention that we see in the individuals from Isla del Coco. We want to work to estimate rates of gene flow and migration across the region, and investigate links between the environment and coral genetic variation. But I want to close by highlighting that this genome skimming approach is efficient and cost-effective for species delimitation and population genomics at whatever level you're interested, whether it be recovering marker genes or looking at fine-scale patterns of population differentiation. And that we can understand now that the Eastern Pacific Pocillopora species are distinct from Indo-Pacific species, and management plans should aim to conserve this unique biodiversity in the region. And I'm very happy to talk more about this with people who are interested in applying these methods to their system. And with that, I'd like to thank everyone who's been involved in this project, especially my Smithsonian Fellowship advisors, and want to highlight my contact information if you want to reach out and ask any questions. My email is connollym@si.edu. Thanks again to the organizers, and I look forward to talking with you and learning more about the discussions you've had at the meeting. Thanks again, and bye-bye.